After the last video, we should have a pretty good idea of the basic processes involved in producing an action potential. But just to be safe, I'm going to go over those basics one more time, but this time with some different visuals and a little bit of different wording, just to see if I can help some of that information sink in. Then once we've done that, I'm going to start building on our base knowledge and start talking about what happens before an action potential can occur and how that signal gets transmitted onto the next cell when we reach our axon terminals. So lots of stuff coming up in today's video. Let's start by orienting ourselves on a new image here that's showing our cell membrane. So we have our phospholipid bilayer here with the yellow, where the yellow is the phosphate heads, and the two little tails there, those are going to be our fatty tails. So on the inside here, this is the intracellular fluid, meaning it's on the inside of the cell. Over here is our extracellular fluid, meaning that's external to the cell. So you should remember that our phospholipid bilayer, the membrane here, doesn't allow things to pass through it because it's both hydrophilic, meaning that the heads like being next to water, so it has a charge. The tails are hydrophobic, meaning they are uncharged and don't like to be near water. So molecules have a hard time passing through both of those properties. They can only usually interact with one or the other, but not both. So we end up with some stuff that tends to be outside and some stuff that tends to be inside, and they can only be moved across the membrane by two different means. And we can actually see both of those means in this image here. The first of which being these sort of flowery looking blue things. Those are proteins, and they're specifically protein channels that allow these ions to pass into or out of the cell when they're opened. We also have slightly different blue things here, and these are our sodium-potassium pumps. These are also proteins, and they move sodium ions through the cell membrane to the outside. So they're going to pump 3 sodium ions outside, and when they sort of cycle back, they're going to bring 2 potassium ions inside. So that's that cycle of 3 positives going out and only 2 positives coming back in. And this process, this pump, is what drives us having a more positive charge outside, and therefore having the inside be comparatively more negative. We can also look at a little bit more detail about our ion channels, or protein ion channels, depending on how you want to call them. They're made of proteins and they move ions, so they're usually called something in the family of ion channels or protein ion channels, depending on who you ask. So those are those gated pores, tunnels through the membrane. They're made of proteins, and they limit which ions flow into and out of the cell. And so they can be opened and closed. Generally, they sit closed so that nothing can pass through, but they can be controlled by one of two means. Some of them are going to be chemically gated, which means that neurotransmitters or hormones can turn them on, can open them up. And some of them are electrically gated, meaning that a change in the electric potential of the membrane can cause them to open up. So that quality is what allows an action potential to happen, where a slight change in the electric potential of the membrane can cause all of the channels to open, all of a specific type of channel to open. In keeping with the idea of introducing sort of additional concepts that relate to what we'd already talked about, I want to talk about a second kind of potential. So we've already discussed action potentials, which is where the cell fires, and we talked about it being an all or none response, where as soon as we cross that threshold, we're going to have that full cell response. But we didn't actually discuss how we get to that threshold point. And so there's actually a specific term for what happens in that sort of pre-action potential stage. So we can talk about what's called a local potential. And a local potential is going to be a partial depolarization. So instead of where with a full action potential, we will depolarize the cell membrane at that point, and we'll have the charge of that area change from the -70 or so up over 0. In the case of a local a local potential, we're only having a slight depolarization. So usually just a couple of millivolts more positive, so shifting towards zero. And this is usually triggered by either, actually, from our previous slide, either neurotransmitters or something, or other electrical charges opening a couple of our sodium ion channels. So having that local trigger, which is usually a signal from a neighboring neuron that's sent over some neurotransmitters or something, that can cause a local potential. And each individual signal is not usually enough to cross over that threshold. Usually you need quite a few of those local potentials all together in the same place in order to get enough of a depolarization built up that we cross over that threshold and trigger a full action potential. And just from how I'm talking about this local potential, you might have figured out that it's very different in terms of characteristics from our action potentials. So where our action potentials are an all or nothing, where as soon as they start, they're always going to occur of the same shape and size and magnitude, a local potential is what we call a graded potential. And this means that it varies in magnitude with the strength of the stimulus that produced it. So if more neurotransmitter signals come in, then we'll have more of a shift towards zero. And if we have multiple signals from, say, different neurons coming in, we can have more and more of these local potentials building together, adding together, and they might then push over our threshold to trigger a full action potential. So we can say that our local potentials are graded potentials, so they can build up. They're proportional to whatever force caused them. And in contrast, our action potentials are non-graded. So that's where that all or none rule comes in. So up until we hit that threshold, we can sort of gradually build in magnitude. But once we cross that threshold, we have a full response no matter what. And just to show an image on the other side, this one's from the textbook. So we show here the voltage across a cell membrane, and here the time in milliseconds. So we can sit at our resting potential. We can have an input stimulus, so usually an incoming signal from a neighboring neuron. We might have these failed initiations. So these would be local potentials that didn't have a large enough magnitude to bump over our threshold. But if we do have one that's sufficient to hit that threshold point, what we'll do is then trigger that action potential. We have that all or nothing trigger point, and a full depolarization occurs, the action potential fires, and then we go through that process where we repolarize, go through the refractory period, and then reset. So if our local potential manages to hit or exceed that threshold, it's then going to change the sort of the electric potential in the membrane at that location enough to open up all of our electrically gated sodium ion channels. So once we've changed the electric charge enough, they'll all open spontaneously in that area, and that's what causes the action potential to occur. And so here's just another image that might actually look a little bit more accurate to what we would expect to see, where at that resting potential, we'd have sort of a slow increase, and that would be caused by a bunch of different local potentials adding together. And then once we hit that threshold, that's when we've triggered all of those sodium ion channels to open up and allow our sodium to flow inward here. Now, you might have noticed that between these graphs, there's sort of differences in terms of what numbers they have on the axes. And the reason for this is because different cells, so different neurons, will actually have different resting potentials, different thresholds, and even different peaks for their action potentials. So it all depends on the qualities or the traits of each individual neuron. We typically talk about a minus 70 resting potential, a positive 30 or so peak, and then the threshold is usually between 10 and 15 millivolts up from our resting potential. But just keep in mind that those numbers aren't absolute. They don't apply to all neurons. We're just giving a general picture. So if you're wondering why the axes don't all line up exactly, that's why. And just to go over our talk of the difference between our action potential versus the local potential of being graded or ungraded, I have another slide here that talks about the steps or the sort of topic of depolarization. And so we know that our action potentials are that abrupt depolarization of the membrane. It allows the neurons to communicate. That's that big upward peak. And we've said that our action potential is ungraded. That means that unlike the local potentials, where the magnitude can build up, where it changes based on the input signal, with our action potentials, as soon as we hit that threshold, all of the sodium ion channels open no matter what. So there's no difference in magnitude, there's no difference in size of response, and that's because of that all or none law. So it's going to occur at full strength or it's not going to occur at all, and whether it occurs or not depends on if those local potentials are enough to push the charge of the membrane above that threshold. So we might say also that an action potential is non-decremental, which means that it's going to travel along the axon without any decrease in size. So this is actually the trait that allows action potentials to transmit that signal along the full length of an axon, no matter how big the cell is. So because each successive action potential traveling along the surface of our axon is going to trigger the next and the next and the next area, they're always going to fire at full amount. They're always going to let all of the sodium ions into the cell to sort of move things along. So that's what's happening when it's non-decremental. So the signal doesn't get lost over distance. An interesting note here is that our local potentials, the ones that sort of either build up or don't, if I actually hop back to the graph that showed them here, these are decremental. So it means that if they occur and they don't trigger an action potential, if they don't hit that threshold, they will just taper off. So they don't travel indefinitely across the cell. They aren't strong enough to propagate down the whole cell. That's why we call them local potentials, because they only work in one localized area. And so our action potentials are the big ones. They're non-decremental, they don't decrease in size, and they're going to propagate at the same size, at the same magnitude, all along the cell, beginning to end. So those are the traits that we want to keep in mind for the difference between our action potentials and our local potentials and how they relate to one another. And because I know that this is a complicated process, and I know that there are lots of moving steps involved, and sometimes this doesn't sink in immediately, I've done two things. The first is I've included some videos on our e-class that should help you see or visualize some of these moving steps with animations. And I find sometimes having moving pictures can help illustrate some of the concepts a little bit better. The second thing I'm doing is what I'm talking about right now, which is going over it again, giving you slightly different visuals, giving you different exposure to the same concepts, but maybe with a little bit of a different explanation. So if you already understand action potentials, this may have been a lot of repetitive, boring information, but if it hasn't sunk in quite yet, hearing it again and hearing it in different ways helps you absorb that information and retain it for the future. So that's why we're doing this. And on that note, I've actually borrowed an image from a different of my psychology textbooks because it does a really good job of breaking down what's happening at each of these stages in the action potential chart with what's happening at the level of our ion channels. So if we break this down here, when we're at our resting potential, when we're just chilling and the cell is at rest, our sodium channels, the red ones, are closed, and the potassium channels, the blue ones, are also closed. So the membrane isn't allowing things to travel across it. We're sitting at our resting potential. So the inside of the cell is comparatively more negative than the outside of the cell, and that's fine. Then we have a local potential. And so when a local potential is happening, it's triggering just a couple of these sodium channels to open up. So a couple of them will start to open. If enough of them are caused to open by sort of strong enough local signals, then we can hit that threshold. So if our local potentials reach the neuron's threshold, if we can hit that minus 60 or so line, then it's going to trigger our electrically gated sodium ions or ion channels here. So the red sodium ions can rush through the now opened sodium ion channels, and that's going to bring lots more positively charged ions inside the cell. So that rushing in of sodium ions is what's causing this increase in electrical potential here. So then once we hit the point where a lot of these sodium ions have come in, we have a switch over where the sodium channels are going to close and the potassium channels are going to open. So potassium ions, the blue ones, are also positively charged. So when the cell is now full of lots and lots of positively charged sodium ions, the potassium want to get away from them. They'd like to get away from the positives. They want to go outside. So when our potassium channels open, the blue potassiums can flow outside of the cell, and as those positive charged ions flow out, we see this drop off in our electrical potential because the inside is now getting comparatively more negative as those positives move outside. And this process, this flowing out, can actually take the cell more negative than it was at the beginning. So it's like the momentum of all of those ions leaving continues too extremely. It takes us past our resting potential, and then we end up being too negative for a little while. So this is when we're into our refractory period, where the cell is more negative than it was, and therefore it can't fire again. So at that point, our potassium channels are going to close, but we're now at a point where all of our potassiums are outside and all of our sodiums are inside, and that's not how the cell likes to sit. So to get back to its resting potential, to get back to where it wants to be, those sodium potassium pumps are going to turn back on and switch or trade the sodium that are inside and put them outside, and then bring the potassium that are outside and move them back inside. So the pumps activate, move everything back to where it should be, and that pump process is what's bringing us back to our resting potential here. So that's just another walkthrough of that process. And I did mention a refractory period where it's impossible for the cell to fire. We've only so far talked about one kind of refractory period, and this is the absolute refractory period. So this is when our sodium channels don't respond to any further stimulation. This is basically saying that the sodium ion channels had closed when we started pumping, or when we started allowing all of the potassium out of the cell. And once those are open, so in this stage here, the sodium channels are ignoring all of the signals. The potassium channels are open through here, and even down here, they start to close up, but the sodium channels, the red ones, are still not receptive to any kind of signal. So even though they are, or they have the potential to be gated channels where we can open them up, they're ignoring any signals during this period here. However, there's a second sort of extra step that happens after this absolute lockout. So there's something called a relative refractory period. And this is when we're still a little bit negative. We're still sort of in this stage here. So the cell is still hyperpolarized. It's more negative than it should be. But the sodium ion channels could respond to an electrical signal. They could respond to one of our incoming signals if the signal was strong enough. So what's basically going on is a new action potential could occur if we had a really, really, really strong local potential, but it would have to overcome a lot of resistance because the cell is more negative. So most of the time, we really care about this absolute refractory period where you physically cannot have a new action potential that cannot occur because the sodium ion channels will not open up. But then the relative period is where if we had enough of a signal, if we had a strong enough incoming signal, we could trigger a new action potential. And this difference here between our absolute cutoff and the relative cutoff tells us that if we have an extra strong stimulus that we need to pass along, our cells or our neurons can fire just a little bit faster if the signal is strong enough. Now that's a bunch of information or new information about action potentials, but that's all we're going to add to that part of the discussion. What we're going to do now is start looking at some of the other parts or the other features that we saw on our neuron diagrams that we haven't actually addressed yet. So some diagrams will show what's called an axon hillock, and this is basically the point where our axon joins with the cell body or the soma. So some diagrams will indicate that for us. It matters a lot more when you get into some of the nitty-gritty about how cells fire. For us, we're just going to care that that's the part where the axon connects to the cell body. The next thing that we can point out is the myelin sheath. So myelin is a fatty, whitish insulation layer that's derived from glial cells during development. So this is going to wrap around our axon and basically act as insulation. So if you think of the plastic coating on an electrical wire, how it prevents the electricity from seeping out, these pieces of myelin sheath, this myelin, isolates the axon and all of the stuff that's going on inside it from the rest of the environment. It also does something really cool because we have these gaps between each of these sort of bundles of myelin sheath, and these gaps are called nodes of Ranvier. And so that's a place where the myelin sheath is very thin or even absent. And where it's open like that, that's where the cell's surface, the cell membrane, is exposed to the environment. So what happens is because myelin is sort of tightly insulating the axon in between these points, we can't have action potentials generated underneath the myelin. So the cell doesn't even have, say, protein channels or ion pumps under there. They don't need them because with that insulation, it allows those charges, it allows the signal to transmit and jump to the next gap. So at that next node of Ranvier, we have a gap, we have exposed cell membrane, and at that point, we have ion channels that can then be opened and pumps that can reestablish that balance. So we have action potentials sort of jumping from node to node to node. So it would go from here, and then there's no sort of new action potentials until we get here, and then we have new action potentials generated, and then we hop ahead, and then the new ones go here, and so on and so forth. And this is actually a really efficient way of doing it. First off, because it's going to save time, because we don't have to go through the process of an action potential firing at each and every single point along the whole length of our axon. Instead, they only have to fire at the points where there isn't myelin. So it can fire here and then skip over all of this stuff and fire here and skip and fire. So we save lots of time. And that actually allows signals in our neurons to fire much faster than they would otherwise. This also saves us energy. Because as you can imagine, if we are pumping ions sort of in and out of the cell and setting up these gradients and then having all of that function, that would be using up a lot of energy in the cell. So if we don't have to generate action potentials anywhere that the myelin is covering, then we get to save that energy as well. So the myelin sheath, this extra insulation, lets us move faster, it lets us save energy, and it lets us get our signals where they need to go a lot quicker without interfering with other parts of the cell. The next thing that I want to mention very briefly is glial cells. We don't get into a ton of details on glial cells other than the fact that they sort of put the myelin sheath in place. They're also there to help support and surround our neurons. So they guide neurons into the right places as they develop. They hold them in the right place. They get rid of debris. They bring in nutrients. So glial cells are a fantastic support network for our neurons. But we don't get too much into them because we could spend a lot of time talking about all the different kinds of glial cells. So I'm just acknowledging them here so that we can move on. Okay, so we've spent all this time talking about how action potentials occur and what's going on within a cell, but we've only hand-waved the fact that this communication happens between cells. So how does the action potential's information get from one cell to the next? So first, I'm going to introduce a little bit of terminology here. So when we have, actually, let me grab a pen here so I can draw, because I find that a little bit easier. So if we have our neuron, and it has the axon and the terminals at the end, and then it's probably got some dendrites over here. So the signal is always going to be moving in this direction. It's always going from the dendrites to the axon. And so once we get to the terminals, the axon terminals, at the very, very bottom, we have what are called terminal buttons, which are basically just these little protuberances. They look like little balls on the end of the axons, and that's just to increase surface area to help us transmit the signal better to the next cell. So the next cell is going to be another neuron. It's going to have its dendrites lined up sort of over here, and then it'll have its axon and then its own terminals down there. And so we're going to be looking at this part in the middle where we jump from one cell to the next. But I just wanted, as I was saying, to introduce some terminology. So this gap is called a synapse. So the terminal buttons of this axon don't actually touch the dendrites of the next neuron. They come very, very close together. There's a small gap here, and that gap is called the synapse or the synaptic cleft. So it's just a fluid-filled space that exists between those neurons. If we wanted to talk about the neuron that comes ahead of the synapse, we can call this the presynaptic neuron, because it's the one that's in front of our synapse as the signal flows. This second neuron, the one whose dendrites are at the synapse, that would be our postsynaptic neuron. And that's because it's the neuron that follows the synapse as that signal travels. So if we talk about a presynaptic neuron and a postsynaptic neuron, we're just talking about the order they come in this signal process. So when we're looking at this diagram here, and we have the axon, and specifically the terminal button of that axon here, that's the axon and the terminal button of the presynaptic neuron. Then we have the synapse, which is the gap between the two neurons. And then we have the dendrite, these parts of the postsynaptic neuron. So the neuron that's after our gap. And so our signal is transmitting in this sort of a direction, but this is where we're going to start talking about how do we go from an electrical signal, which was our chemical charges in the axon, and then we're going to actually look at turning it into a chemical signal to go between the cells, and then we're going to turn it back into an electrical signal once we're in the next neuron. So it goes electrical, then chemical, then electrical. And we've already talked about the electrical part because that's our action potentials. So now we're going to zero in and focus on this chemical part and start talking about neurotransmitters, which are the chemicals that carry the message from our presynaptic neuron to the postsynaptic neuron. And then just one more thing I want to point out while we're looking at this image here, we have these blue circles. These are called vesicles, and they're basically sort of bubbles or sacs that contain our neurotransmitters. So they have lipid membranes the same as the cell does. And then what they can do is if they come up to the cell membrane, they can bind with it and open up to dump their contents from inside the vacuole. They'll then dump it outside into the synapse. So that's the basics of what we're going to talk about here. All right, so let me get rid of our visuals there, and then we can hop to the next slide. So when we're talking about our neurotransmitters, I already kind of gave the definition, but they're chemical substances. They're going to carry messages across the synapse, and they can serve one of two functions. They can either excite the next neuron, or they can inhibit the next neuron. So exciting, or being an excitatory neurotransmitter, means that you make it more likely that that next neuron fires. If you inhibit firing or are inhibitory, you make it harder for the next neuron to fire. So if we think of our sort of local potentials, if we're going to be more likely to fire, then we are going to increase, sort of repolarize that membrane. Sorry, I said I mean depolarize. Let me hop to the image that I'm thinking of. So with our excitatory neurotransmitters, and I'm going to grab my visuals because I find these help. So we have our sort of cell at its resting potential. So this is our postsynaptic cell. And so these neurotransmitters are what are creating those local potentials. So if we're making it more likely for the next cell to fire, we are making this cell more positive. We're getting it closer to that 0 mark because it starts at negative something, right? So when we're depolarizing this membrane, we're causing that increase in charge, hopefully enough to hit our threshold. And if we get enough of these excitatory neurotransmitters, if they cause large enough local potentials, they're going to trigger our action potential. So it increases the likelihood of an action potential because it's reducing the polarization or making the cell membrane more positive. If instead, get rid of that, if instead we're looking at inhibitory neurotransmitters, can change colors to make it a little easier, they're going to be making the cell more negative. So we can talk about it hyperpolarizing the membrane. So the local potentials, instead of getting higher, are making the charge even lower. So in order for this cell that's been inhibited to fire, we would have to get a ton of excitatory signals to bring it back up just to hit the threshold before we could fire. So by making it more negative, by hyperpolarizing that membrane, we make it less likely that an action potential is going to occur. We're making the cell more negative and making it harder for that cell to fire. So just another way of saying it, excitatory neurotransmitters increase the probability of a neuron becoming active or firing, and inhibitory neurotransmitters decrease the probability of a neuron becoming active. And these are just a couple of examples of some well-known excitatory and inhibitory neurotransmitters. I will point out that dopamine can do both, which is just lovely and confusing. But depending on what kind of site these neurotransmitters bind to, they can create different effects. That's one of the things that allow us to have such complex signals in the brain, is that we have different neurotransmitters that can be released from different places, but we also have different receptor sites that they can bind to, and when they bind, they can create different effects, which is how dopamine, if it binds to a receptor that creates an excitatory situation, it can cause that increase in the charge so it can get closer to 0. Or if it binds to an inhibitory receptor, it can sort of cause it to be more polarized. It can go in the opposite direction. And just to talk about how our neurotransmitters might bind to receptors, we have another visual here. And again, I'm sort of pulling things from multiple different books because I like different visuals to illustrate different points. So this does a great idea of showing us this presynaptic neuron. They're also calling it a sending neuron, which might help you visualize. And our postsynaptic neuron, the one that's receiving a message. And then we're going to blow up this synaptic space, our synapse, where we have our presynaptic axon terminal, or specifically the little bulb on the end being those axon buttons or terminal buttons. And then we have the postsynaptic membrane, so this would be the dendrites of the next neuron. And so we have our vesicles. They're going to move sort of towards the end, and when they're triggered, they can bind to that cell membrane and dump the neurotransmitters out into this synaptic space. We're going to talk on the next slide about the receptors, but just the basics of it is that different neurotransmitters are going to be different shaped molecules. So they've simplified it here with balls with different sized and shaped sort of pointy bits on them. That's kind of the same idea. These molecules will all have different shapes, and they'll be able to bind to different receptors. So if this neurotransmitter fits this receptor, it can bind and activate that receptor, triggering whatever the effect is on the other side. So maybe it's going to open up some sodium ion channels and cause an increase in the charge. It's going to depolarize that membrane. But if the neurotransmitter doesn't fit the receptor, then it can't bind there, and then it doesn't activate it. And if we want to look at the process of sort of setting this thing up, you usually start with the synthesizing your neurotransmitters. So the cell will be set up to assemble them, put together all the moving parts, and then they store them in these synaptic vesicles. So these little sort of bubbles that contain those neurotransmitters. The next step is to release them out into that synaptic space, out into the synapse. And then if they find receptor molecules they can bind to, they'll bind to those receptor sites. And then once they've bound, there's usually a final step that involves cleaning up. So you don't want these receptors always on, so you want to deactivate them. So you can either re-uptake your neurotransmitters. So there are special systems in place where things will go around and collect the neurotransmitters and put them back into vesicles to be reused, or they might break them down. So you might see an enzyme that comes along and maybe cuts up all of the neurotransmitters so that they can be recycled and turned into something else. But in either case, those deactivate or disconnect our neurotransmitters so that they only act in a very specific area for a very specific amount of time. And just another example of what we're thinking of with our neurotransmitters binding to a receptor. This is from our textbook, and it shows that the neurotransmitter can sort of fit into a slot, kind of like a lock and key. So the key is like the neurotransmitter. If it has the right shape, it will activate or bind at that receptor site and be able to turn it on or open the door. So that's kind of how we think about it. And then very, very briefly, I just want to talk about how drugs can start imitating or acting like neurotransmitters, or at the very least, affecting how our neurotransmitters function. And they tend to do this by activating the same receptors that our neurotransmitters do. So drugs can be created that either replicate or prevent the action of our receptors. So if this drug is what's called an agonist, actually I have it on the next slide, so we're going to talk about the terms sort of interchangeably between the two. So if the drug is an agonist, then it's going to replicate the receptor action. So what that means is when it binds to that receptor, it's going to behave as though the neurotransmitter that belongs there has bound, and it's going to do whatever it was supposed to do. So if this is supposed to be a dopamine receptor, and we have a drug that comes in and binds as if it was dopamine, and this site opens or starts activating as though it was bound by dopamine, then we can call that an agonist. We can also have what's called an antagonist, and that's when the drug prevents the receptor from acting. So what happens there is that the drug would bind and sort of prevent dopamine from doing anything. And it can work in two ways. It can either be direct or indirect. So I've been kind of pointing at our competitive or direct competition. So that's when the drug binds to the neurotransmitter's binding site and either activates it if it's a direct agonist, or it binds to that site and turns off this receptor. That would be a direct antagonist. So we could even draw in the fact that it turns it off. It doesn't act the way that it should. And then if it's an indirect competition, then we have a neurotransmitter site, but the drug can bind elsewhere. So if it's an indirect competition or non-competitive binding, then it's not going into the same site. It's going to bind somewhere else on that molecule. But if it's an indirect agonist, if it binds to that other location, it can still turn on our site. It can turn on our receptor. And if it's an indirect antagonist, then it's going to bind somewhere else and it's going to turn off or prevent this neurotransmitter from making this active. So that's how drugs can either sort of imitate and act as though they are our normal neurotransmitters, or they can act in a slightly different way and just cause similar effects by activating or deactivating the same receptors. And again, you can take entire courses on drugs and neuroanatomy and stuff like that, but we just keep it very, very brief for this course. So we can leave it there for this topic. And then next, we're going to start talking about how we organize all of these neurons together to create our nervous system.